Welcome to the PubMed Daily Review for April 14th, 2026. Today we scanned 5,622 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, Precise Gene Editing of Pathogenic Lamin A Mutations Corrects Cardiac Disease by Serd M. Caravaya and Eric N. Olson from A Department of Molecular Biology, Hammond Center for Regenerative Science and Medicine, University of Texas Southwestern Medical Center, Dallas, Texas 75390. Let's break down its key contributions. Laminopathies, caused by mutations in the Lamin A gene, are severe genetic disorders affecting the heart and muscles. Unfortunately, current treatments are limited. This research investigates whether precise gene editing can correct these disease-causing mutations and restore normal function. The researchers developed precise base editing tools, AB and CBE, to target specific Lamin A mutations. They tested these tools in human heart cells derived from stem cells and in mouse models carrying these mutations. The goal was to see if correcting the gene could reverse the disease-causing effects observed in these models. The results were highly encouraging. Gene editing successfully corrected the cellular problems in heart cells carrying the mutations. Furthermore, when delivered to mice, the base editing approach prevented the severe cardiac and muscle disease symptoms and significantly improved their lifespan. This demonstrates a powerful therapeutic potential. This study shows that precise gene editing is a viable and effective approach for tackling devastating genetic heart and muscle diseases caused by Lamin A mutations. It opens a new avenue for developing therapies for laminopathies. While these findings are exciting, further studies will be crucial to confirm safety and efficacy before human trials can begin. Up next, our second paper out of 5, Functional CRISPR Screens Define Genetic Drivers for Cancer Transformation and Progression from Non-Cancerous Cells by Shixin Ma and Tung Fei from Key Laboratory of Bioresource Research and Development of Liaoning Province, College of Life and Health Sciences, Northeastern University, Shenyang 110819, China, SM-YL. Let's break down its key contributions. Cancer development is a complex journey, starting with normal cells acquiring genetic changes. But what are the very first genetic switches that flip a normal cell towards malignancy? And do these switches work the same way across different cancer types or environments? This research dives into those fundamental questions to understand the earliest steps of cancer. To pinpoint these initial drivers, the researchers used powerful CRISPR gene editing technology. They screened for genes whose loss could transform normal mouse and human cells into cancerous ones. These screens were conducted in various settings: simple 2D cultures, more complex 3D structures, and even within living mice. This allowed them to see which genetic changes consistently promoted transformation and which ones were specific to certain conditions. The study revealed that TP53 and NF1 were like universal keys, consistently unlocking cancer transformation regardless of the experimental setting. However, the majority of other genetic drivers identified were highly specific, depending on the species, tissue, or microenvironment. Despite this variability, all these drivers ultimately converged on key cellular processes like epigenetic control, metabolic shifts, and growth signaling pathways. This research offers a novel perspective by focusing on the genetic barriers that normally prevent cells from becoming cancerous. It emphasizes that cancer drivers are not one-size-fits-all, with significant differences between species and environments. Ultimately, this work provides a crucial framework for understanding the very first steps of cancer development and could pave the way for new strategies to detect or intervene in early-stage disease. Up next, our third paper out of 5. AI-enabled flexible sensing ecosystems for Parkinson's disease, advancing digital biomarkers and closed-loop interventions by Jiadong Jin and Li Jiapan from Collaborative Innovation Center of Advanced Microstructures, Jiangsu Provincial Key Laboratory of Photonic and Electronic Materials, School of Electronic Science and Engineering, Nanjing University, Nanjing 210093, China, JJ. Let's break down its key contributions. Parkinson's disease is the fastest-growing neurodegenerative disorder, affecting millions worldwide. The classic motor symptoms like tremors and gait issues significantly reduce patients' quality of life. However, managing PD is challenging because clinical assessments are like snapshots, missing the day-to-day reality of symptom changes. This makes it hard to truly personalize treatment and manage the disease effectively. This review explores a new paradigm for Parkinson's monitoring: flexible sensors combined with artificial intelligence. Instead of bulky, uncomfortable devices, we're looking at skin-conformant electronics that can capture subtle movements and biosignals. These sensors work hand-in-hand with AI algorithms, including Edge AI for on-device processing, to turn raw data into meaningful digital biomarkers. The goal is to create a seamless ecosystem for continuous, personalized home care. The research highlights significant advancements. Flexible sensors have achieved impressive milligram-level sensitivity, a crucial step for detecting subtle PD symptoms. However, a notable translational gap exists, meaning many innovations are still limited by small studies and lack of real-world validation. The integration of AI, particularly multimodal fusion, is vital for accurately translating sensor data into clinically relevant digital biomarkers. Furthermore, using Edge AI allows for private, real-time feedback and interventions directly from wearable devices. This integration of flexible electronics and AI is truly transformative, moving us from reactive clinic visits to proactive, personalized digital care for Parkinson's patients. The next crucial step is to bridge the gap between lab findings and real-world application by conducting larger studies and ensuring regulatory approval. Ultimately, continued innovation in AI and sensor technology will pave the way for highly adaptive, closed-loop interventions that can significantly improve the lives of those living with Parkinson's. Up next, our fourth paper out of 5, Enterococcus-driven metabolite-host gene networks in IBD-associated colorectal carcinogenesis. Integrative multi-omics and experimental validation by Gang Huang and Yuan Liao from Department of Laboratory Medicine, Guangzhou Cancer Hospital, Guangzhou, Jiangxi, China. Let's break down its key contributions. We know that long-term inflammation in the gut is a major driver for developing colorectal cancer. While the gut microbiome is implicated, the precise role of specific bacteria like Enterococcus is still being uncovered. This study dives deep into how Enterococcus and its metabolic byproducts might influence the progression from inflammation to cancer, aiming to shed light on crucial molecular pathways. To tackle this complex question, the researchers employed a multi-pronged approach. They integrated vast amounts of genomic and metabolomic data from public databases, alongside experimental validation using cell cultures and advanced single-cell sequencing. This allowed them to build intricate networks of how Enterococcus-driven metabolites interact with host genes and to pinpoint specific genes and pathways involved in CRC development. The study revealed that Enterococcus faecalis significantly boosted the expression of protective genes like IL-10 and IL-1B in CRC cells. However, the story gets more complex. Its metabolites, agmatine and levodopa, were found to markedly induce the expression of MAPK3, a gene associated with poorer survival in CRC patients. This highlights a dual, context-dependent role for Enterococcus in cancer progression. This research underscores the intricate relationship between gut bacteria, their metabolites, and colorectal cancer. The identification of specific metabolites driving pro-cancerous gene expression opens doors for novel therapeutic strategies. Future research could explore targeting these metabolite-host gene networks, potentially using compounds like estradiol or sodium arsenite to better manage IBD-associated CRC. Up next, our fifth paper out of 5, discovery of the SARS-CoV-2 Papain-Like protease inhibitor MR1-114. From structure-based design to in vivo antiviral efficacy by Mulla Reddy Ganarapu and Rui Xiong from Department of Pharmacology and Toxicology are Kinkoy College of Pharmacy, 8041 University of Arizona, Tucson, Arizona, 85721, United States. Let's break down its key contributions. The COVID-19 pandemic continues to pose a global health challenge, with new variants emerging regularly. While vaccines and some antivirals are available, there's a constant need for new therapeutic options. The SARS-CoV-2 virus has a critical enzyme called PIL-PRO, which is vital for its replication and for evading our immune system. Targeting this enzyme offers a promising avenue for developing new antiviral drugs. To tackle this challenge, researchers employed a structure-based drug design approach. They used detailed knowledge of the PIL-PRO enzyme structure to guide the creation of new inhibitor molecules. Through iterative medicinal chemistry optimization, they aim to enhance potency, improve drug-like properties, and ultimately achieve antiviral activity in a living organism. This involved a battery of tests, from enzyme inhibition assays to studies in animal models. The optimized compound, MR1-114, showed impressive results. It potently inhibited the PeelPro enzyme with an IC50 in the nanomolar range. Crucially, it exhibited high oral bioavailability in animal studies and preferentially accumulated in lung tissue. In a mouse model of SARS-CoV-2 infection, oral administration of MR1-114 demonstrated therapeutic efficacy comparable to nirmatrelvir, significantly reducing viral replication and preventing weight loss. This study highlights the success of structure-guided design in developing a novel class of SARS-CoV-2 PeelPro inhibitors. MR1-114 represents a promising candidate with potent antiviral activity and favorable pharmacokinetic properties, including oral bioavailability and lung targeting. These findings underscore the potential of targeting PeelPro as a therapeutic strategy and pave the way for further development of such inhibitors as potential new treatments for COVID-19. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.